The helicase enzyme opens up the DNA strand into two separate strands. It travels from the origin of replication through the DNA strand, breaking hydrogen bonds in the process. The primase enzyme lays down an RNA primer along the open strand of DNA. The DNA polymerase enzyme builds the DNA strands. It starts with the primers and matches new nucleotides to the complementary nucleotides on the old strand. DNA polymerase also takes out the primer and replaces it with DNA. The ligase enzyme connects any ends of the DNA fragments that aren't connected. DNA replication occurs during interphase, specifically in synthesis. At the beginning of DNA replication, an enzyme called helicase opens the two strands of DNA from the origin of replication. The helicase breaks weak hydrogen bonds, allowing for the strand to be separated easily. Next, RNA primase lays down RNA strands in preparation for the DNA polymerase enzyme. Without the primer, the DNA polymerase would not be able to lay down DNA and create a new strand. After the primase, DNA polymerase reads the strand in the 3 to 5 direction, meaning it places the nucleotides in the 5 to 3 direction. Each strand is anti-parallel, so the strands are going in opposite directions and are complementary to one another. However, a problem occurs with the DNA polymerase laying down in one direction. While the DNA polymerase following the helicase can continually lay down the nucleotides, the DNA polymerase on the other strand will have to keep going back and forth since it's laying in the opposite direction. This causes this strand to replicate slower and become the lagging strand, as the other strand is the leading strand. Once the DNA polymerase is finished laying down nucleotides, it will replace the primer with the complementary nucleotides. After the DNA polymerase is finished, an enzyme called ligase will run through the DNA strand and connect any DNA fragments that aren't connected. The new strands are semi-conservative, meaning that each strand is made of the original strand and a new DNA strand. The strand will now continue through cell division.